Hi, my name is Marie and I'm a graduate student working in Victoria D'Souza's lab at Harvard University. Today I'm going to be talking to you a bit about fluorescence microscopy. Now, before we get into the nuts and bolts of fluorescence microscopy itself, I want to first go over what fluorescence is. Many of you may already be familiar with the concept of fluorescence. The most simple example of fluorescence would be the way a jellyfish glows a fluorescent green. On a chemical level, we can illustrate fluorescence using an energy diagram, where we here have on this axis energy, where we start from low energy going up to a higher energy. The way fluorescence works is you take an electron, which starts at the ground state level. I can excite this electron with a little bit of light, which I'm depicting here in blue, and this will cause this electron to jump from the ground state level all the way up to some excited state level. We consider this to be absorption, since it's absorbing light energy in order to go from the ground state level to the excited state level. Now, this electron in the excited state level isn't stable, and so it's going to want to come back down and relax back down to the ground state level. In the process of doing so, it first loses a little bit of energy as a form of energy we call vibrational energy, before it's able to relax back down to the ground state level, coming back down here. In relaxing back down, similar to how we absorbed light earlier to go back up, the opposite is true here, where in order to relax back down, we have to emit a certain light energy. And so this is what we consider emission. This emission here gives us the phenomena of fluorescence. So in the example, the jellyfish we used earlier, the jellyfish absorbs blue light and then emits green light. And that green light is what you end up seeing. We can also depict this using a different kind of diagram. Here we've shown this using energy, but another way to show this is using wavelength. Now, a lower energy, which we see here in the emission spectra due to having lost a little vibrational energy up here, a lower energy corresponds to a longer wavelength. So if I use a different kind of diagram here, I can plot my wavelength on the x-axis with some arbitrary intensity value on the y-axis. As I mentioned before, in my example, I'm absorbing using blue light, and so I can draw a curve representing the possible wavelengths at which we could absorb to excite this electron. This absorption is going to correspond with some emission, and as we mentioned before, the emission will be at a lower energy, which corresponds to a higher wavelength. So we'll see the emission coming out over here. The optimum level of fluorescence would be if you're able to excite at the highest possible intensity of your excitation wavelengths, which will allow you to emit at the highest possible intensity for your emission and give you the strongest light production from your fluorescence. Fluorophores are chemical compounds with the ability to fluoresce. We just went over one example of a possible emission spectra, but different fluorophores will differ in the specific wavelengths in which they absorb and they emit. And this will allow different fluorophores to be able to give off light in different colors, which can be detected independently of each other. Fluorophores can range from small organic molecules, such as this, to larger proteins, such as GFP, green fluorescent protein. The small organic molecules are on the order of 1 kilodaltons, while GFP, green fluorescent protein, would be much larger, on the order of 27 kilodaltons. In this talk today, we're going to be focusing a little bit on GFP because it's probably the most well-known of the fluorescent proteins, and it's a very widely used tool in molecular biology. GFP was discovered in around the 70s, and since then it has been turned into a pretty ubiquitous and versatile tool. Different derivatives of GFP have been made that can actually fluoresce at different colors, and GFP is such a powerful tool because it is non-toxic and it's not destructive to the cell. This allows us to be able to image cells without destroying them or altering them. In this first example that I'm showing you here, GFP has been expressed in cells by itself. It has been put next to a promoter that is specific for neurons, allowing it to be broadly expressed within neuronal cells. This enables us to see both, as you can see in the insert, individual neuronal cells, as well as be able to look at the structure of the brain as a larger whole tissue. Other than expressing GFP individually within cells, we can also attach GFP to very specific proteins and express GFP with those proteins as a fusion protein in order to track where those proteins go. In this example that I'm showing you here, we have a cell that you can see in this image where the blue represents the nucleus of the cell. And we've tagged different cytoskeletal proteins with different derivatives of GFP. In green, we can see actin, which forms this broad cytoskeletal network. And in red, we see tubulin, which you can see on the periphery of the cell. One reason GFP has become so powerful is because we're able to attach it to specific proteins to see how these proteins move and where they are located within a cell. The process of doing this is easier and more straightforward than you might think. All I have to do is take some expression vector where I can take a protein, for example, using the one we just talked about, I could have actin, and I would clone in GFP, here represented using this green stripe, either on the N or the C-terminal end of it. Either way is okay. And when I put this plasmid within my cell and express it, I would start at some start codon and end at some stop codon out here, so that every time my actin is expressed, it will have a GFP reporter tag attached to it, allowing me to track the location of every single actin within the cell. What is so powerful about this technique as well is that you can actually detect GFP down to single molecules, enabling me to be able to discern the location of one individual actin. In order to be able to detect fluorescence in our samples, we have to use a special fluorescence microscope. In order to explain how a fluorescence microscope works, I've drawn out here the basic components in all fluorescence microscopes. We'll start here with a light source that emits visible light. Here I'm representing this visible light using multiple different markers to show that it has different colored wavelengths of light. And this visible light comes out, and over here it hits first an excitation filter. This excitation filter has a very important job where it blocks out all of the wavelengths of light that we don't want, only letting through the singular wavelength of light that we want to be able to use in order to excite our sample. This wavelength of light passes through, it hits a dichroic mirror, which I will get to in a second, before bouncing off, going through an objective, and down onto our sample. As we went through before, our sample having now been excited with light, will also now emit a different wavelength of light, here colored in green. This light will come back out, again through the objective, and when it reaches the dichroic mirror, instead of bouncing, it can actually go through the mirror. Dichroic mirrors are special mirrors where they're able to let through certain wavelengths of light while reflecting and bouncing off different wavelengths of lights. This allows us to be able to direct these different wavelengths of light to the locations where we want them to go. The light having gone through this dichroic mirror will come through an emission filter before being detected by our detector. The use of this emission filter is doing something similar to what the excitation filter did, except what it's doing in this case is instead of the excitation wavelength, it's only letting through the emission wavelength of light. This filter is actually quite important. It helps us increase our signal to noise for our microscope because it blocks out all light that may have come from non-specific fluorescence or innate fluorescence in our sample, allowing us to detect only that which comes from the very specific signal that we are hoping to see in our sample. The key takeaway here is that we're not actually visualizing the fluorophore or the fluorescent protein itself on the sample. We're detecting the signal that is emitted by that fluorophore or fluorescent protein. In this way, we're able to find the location of things that are incredibly small and too small to see by using light as a proxy for where they are. So we can detect down to single molecules even though we can't actually see the molecules themselves. I hope you enjoyed this video on fluorescence microscopy. Thanks for connecting with us at LabXchange.